Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bandettini, and today is a very special podcast. It's my honor to be able to talk to Dr. Seiji Ogawa. And most everyone in the field knows who Dr. Seiji Ogawa is. He is the person who discovered bold contrast back in 1989, between 89 and 91. He was developing and establishing bold contrast as the contrast itself. And of course, in 1992, the first papers came along to demonstrate that bold contrast can be used to look at brain activation. I hope you enjoy the conversation as much as I did. Dr. Ogawa is doing well at 90 and still contributing to the field. So I hope you enjoy it. And it's my great honor to have Dr. Seiji Ogawa on this podcast and speaking to us from Japan and to tell us a little bit of, his perspective of the history and how things happened and, his perspective of how things are going now. So I've known Seiji for a long time, all my career. You were on my PhD thesis committee. And
that's right. I remember that day.
So do I. And I remember also, I was a little nervous. I was very nervous, but I also remember that you asked me a question that still I felt was stuck with me because you asked me a specific question like, well, how does deoxyhemoglobin actually distort the magnetic field? What is the mechanism by which it does this? Like, describe that? And I was trying to describe it, and it wasn't quite right, and you didn't quite agree with my answer wasn't insightful enough, and that made me very nervous. But I still think about that from time to time. Do I really understand this or not?
I don't remember that one. Okay.
But, yeah. So why don't we just get started with, obviously, Jim Hyde, and Jim Hyde was my advisor, and he had mentioned he knew you from long before you discovered bold. He knew of you were a student at Stanford. You got your PhD at Stanford. You got your undergraduate at the University of Tokyo. And he mentioned, and maybe it's correct or not, he mentioned that you were working on blood beforehand with electron spin resonance, or is that right? Or I don't, maybe I'm getting the story wrong.
Most of the blood thing is coming with Hardy McConnell. Do you know Hardy McConnell? Remember?
I know the name, yes.
Yeah,
yes.
At Stanford. Jim Hyde at that time, he was at the Varian there.
Yes.
Yeah, right. And he was just about left there on that time. So
that's right.
The reason why we played with hemoglobin, not blood yet, is because of the what you call transition of structure, quaternary structure change.
Yes.
Whether we can detect or not. And McConnell's spin level was the one we are working on it. That's what the ESR on the blood is related to.
Yes.
That's the only thing. Other than that, before that, I did some of the free radical study about the ESR. Actually, at that time, I was involved in the development of radiation damage. Not damage, but improvement by the radiation to plastic material. When those kind of things was still dream at that time. That's what we were working on. And to see free radical is one of the easiest way to look into it.
Yes.
It turns out to be, it was not improving, but destroying plastic.
Okay.
turns out to be. So I left the field.
Okay. Okay.
That's why I went to the Melon,
Okay. Okay.
and where they are doing those radical formation by electron beam irradiation.
Okay.
So
Okay.
yeah, radiation
Okay.
laboratory. That's where I was. I went there, and
Okay. So that was at so you okay. Okay. So you went to Bell Labs.
because one of the big reason is I didn't have any connection to go back to Japan. In Japan, in order to get academic position, you have to have a fairly good solution to the big boss. I didn't
Yeah.
have it anymore because I left the field completely. So,
Yes.
and
Oh, that's
um,
interesting.
yeah,
Okay.
to go to Bell Lab is one of the dream for physics people.
Yes.
And
Well,
so
back
that,
then it was an amazing place. It was
yeah,
incredible.
that's right. That's right.
Yeah.
Unfortunately, that is the only place I can go in order to get my visa status changed.
Okay. Okay.
There were
Okay.
in
That's
any other place, it was not possible there at that time.
yeah.
That's one another reason why I went to the Bell. But
That's
anyway,
okay. Well, that's a good reason. That's a very practical reason. And yeah. And what was it like at? I mean, Bell Labs at the time was like this. I mean, everyone. Once again, this is sort of a legendary place that
that's
uh,
right.
people were doing many different things, and you just went there. And what was it like? How did you decide what to
That's
do?
right.
Uh,
Yes. So Bob
yeah.
Schumann at that time create insisted to create biophysics study at the Bell Lab. That's what. That's why we went. He hired me there. So
Yes. Yes. Okay. All right. So Bob Schulman was there, and
yeah,
okay, and then you were working for a while at Bell Labs, and then and this is really interesting. How did you move into studying blood again and susceptibility effects?
yeah,
What went through your head
yeah. To
yeah.
work on the to work on with the hemoglobin was a topics with Bob Schumann.
Ah, okay.
Yeah. So so we discussed how to deal with hemoglobin for NMR.
Yes. Yes.
220 megahertz NMR was a big instrument at that time.
Yes.
Yeah.
Yeah.
So that's
And
why
you
I started again back to the hemoglobin that time.
okay.
Yeah,
Okay.
yeah,
And it was mostly, I guess, in the context of spectroscopy, looking at line broadening or
yeah,
um,
yeah. At that time, more or less paramagnetic state of the heme, it may influence NMR signal around that heme group. So I
okay.
was quite familiar with the paramagnetic state of hemoglobin.
Yes. Yes.
Yeah,
Yeah. Which is unique. I mean, I was just sort of thinking about that today, that it's one of the only, I mean, this is the beauty of NMR and MRI is that biologic materials like hemoglobin can change their magnetic properties or other properties depending on their physiologic state. And blood was a huge thing. Blood was is the most. I mean, it's amazing how lucky we are that blood
that's right.
changes its magnetic
That's
state
right.
with
Yes.
but how did you actually so I'm it's interesting. So you were sort of working on that, but then when did it occur to you to that, oh, this might, I know in the field there was like John Shank was talking about blood. I remember reading a paper by him or in a book chapter or something saying, oh, hemoglobin might have an effect, but it's not that big, and it's not that interesting, or it's sort of small, and whatever. And I know Keith Philborn and others were doing hemorrhaging, and they're looking at hemorrhage, and there was a contrast. But when did it occur to you to say, hey, this is a bigger effect than we think?
The thing is the following. Before that time with Bob Schumann, you remember those in vivo NMR, so called.
Yes.
right?
Yes.
And looking for
Yes.
metabolites. And then after Bob Schumann moved to Yale, they are working on the various metabolites, and people who are doing a human in the big instrument, the signal is so big relative to the small animal brain. In order to see
Yes.
metabolites, you need a lot of volume to see it, right?
Yes. Yes.
And I thought, well, this is no point working on the metabolites anymore with this kind of. So I didn't want to do anymore. So imaging is the next stage to get into. So
Okay.
I was quite ignorant on the image, but anyway, I wanted to see what kind of signal, image signal is, what that means.
Yes.
I wanted to know what represent those signal, brain,
Yes.
various parts of the brain, whatever thing is T2, T1, T2 things, yes, but what that signal represent to us? That's what my whole
Interesting.
question,
Yes.
starting image to the functional MRI.
Yes. Yeah.
Yeah,
That's interesting that you had that perspective early on. That I mean, we're still doing that today with MRI. We're digging into more the signal, and we're trying to figure out what does that mean in terms of histology or other things, and what contributes to the signal that we can actually pull out. We're still doing that today, and you started that. So you basically put just a rat brain. You were working at 7 Tesla, and you basically just put it in the scanner, and you saw these dark lines, I guess, for the veins, and I guess you were trying to think, well, what's causing that? I guess maybe that was
yeah,
the question. You made that observation, and you had this question, I guess, and you were right on in your intuition that it was blood oxygenation, and that was still such a beautiful experiment of changing the oxygen level, or this changing the level of anesthesia in the rat, and then doing the test tube verification. It was so convincing seeing the penumbra around the test tubes, and then around the veins, and those set of papers. That was just beautiful. It was just, and it was interesting how it came, like you said, from just, so you started with just putting the animal in the scanner, having the image, and then looking at the image with an idea that, okay, with a deep understanding of how the signal might be affected. But it's interesting because so many other people, this is the interesting part about discovering mold, is that people were already working and sort of knowing that, like if you had a brain hemorrhage, that it would affect the contrast. And people saw that. And a lot of other people were sort of understood the effects of susceptibility, but none of them sort of made that connection of, oh, this is really sensitive to blood oxygenation, and it could be used for looking at brain function. So that, I think that was the key with what and it was, yeah, it's amazing how that's how, I mean, I'm just trying to, how discoveries are made. It's there, and then the person who just makes the observation, and then makes a connection, and then tries something else, then and tries to carry that forward. And that was great. That was
yeah,
just,
yeah,
yeah. And so I guess, and so I remember, actually, also I remember reading your papers, of course, and I remember also looking at, like, the first results that I saw were at ISMRM in 1991. Actually, I remember the date, August 12th, 1991, and Tom Brady put up some movies and of brain activation, and at the plenary lecture, and he was from MGH, and he didn't mention which way the subtraction was done. He didn't say that this showed us an increase in visual cortex. And then also reading your paper, I remember reading, there's one part where you speculated that, oh, with increased metabolism, we might see a signal decrease, because before we actually talked to the people who did PET imaging, like Peter Fox, that, oh, no, you have an overshoot in the oxygenation. And I remember looking for my own results. I remember constantly looking for signal decreases, but only seeing signal increases. And then, of course, you're collaborating with the Minnesota group, and showing activation. What was that like? So what happened? So after you did your animal studies, you realized, oh, this is sensitive potentially to function, to brain function. And what did you do at that point? Because you just had rats. I mean, I guess you could have done a rat activation experiment, but then you collaborated with Camille Ugerbill. What was that like?
To be honest, we couldn't do good functional study on the mouse or rat because
Right.
I didn't know the proper anesthetizing condition at that time. So
Yeah. Yes.
whatever neuroscientists doing those things, usually they put in the very strong anesthetizing compound. Just I did, same thing. So we don't see much. Actually, I asked my friend to measure how the blood signal flow changes by activation to the small animal under anesthetized condition. And he actually helped me to see it. We can see it very small anesthetized condition. No hope to see in the animal.
Yeah.
So the human is the only thing you don't need to anesthetize. So that
Yes. Yeah.
was
Exactly.
the advantage you had, right? Probably
Yes.
something to you or MGH people too. Yeah.
Yeah. And so you started talking to. So what made you decide to talk? I guess Camille was the only group that had high field, or they were getting high field. They were in the process of getting. So you felt that it was critical to have or 4 Tesla at the time. And
Yeah. That's right. That's right.
okay.
Should be
And then you
easier,
were.
even than hard.
Yeah. Yeah.
Yeah. Actually,
Um,
when we see that the data we got the first time, Ravi Menon made a very beautiful image picture out of there. The activation follow the dark line of cortical area, smooth, slow, very closely, amazingly closely. So
yes.
that's very good. Yeah.
That was so
That
stunning.
was
And
for Tesla strengths at that time.
yeah, I often wondered why. I mean, I think that the experiment. I mean, we obviously did it at 1.5 Tesla, and MGH did it at 1.5 Tesla, but and why I wondered why Camille wanted to wait until 4 Tesla. I mean, I think the Minnesota group had a 1.5 Tesla. It would have been worth I guess, but the echoplanar imaging was well with Minnesota it was multi-shot, but it was high enough field that the instability didn't matter that much, and it was really nice results. Yeah. Yeah. So what was that like? I mean, what was the feeling? You probably flew over to Minnesota. You were doing the
Yeah,
experiments with
yeah, yeah. Yeah,
and
all over there. Camille, Ravi, and Sanjay,
yes.
and
Yeah.
Kim, and myself. Neuroscientist from the Bell Lab wasn't there. He came a couple of hours later. Before.
Little did he know what an experiment that was going to be. And so people always ask me like, who was the first? Was it MGH or Minnesota? And I don't really know. I think that MGH was in May, and I don't know May of 1991, and I don't know if Minnesota was before or
Uh,
after.
uh,
I
I understand that one of the department head of our biophysics department visited MGH, and the MGH people told her that don't tell Seiji what we are doing there. But she was explained by them what it is. But she said, "Oh no, Seiji is working with Camille over there." So they rushed to write a paper because of that.
Oh, that's great. That's great. So yeah, so I don't I yeah, I think it's probably best to, it's one of those things that, I don't even know. It almost doesn't matter who was first. It was your discovery of bold, and then the next step, and then the next step, and everyone builds on each other, and everyone did it differently. I remember for myself, I was just a graduate student, and I I remember seeing Tom Brady's work, and I didn't know about Minnesota's work, but I think that Jim Hyde, he's like, oh, you have to talk to Seiji about this. You have to talk to Dr. Ogawa. And I remember faxing you some results, not even knowing that I actually didn't know that you were collaborating with Minnesota on this. And you wrote back saying, oh yeah, this is very interesting.
I
And
was very lucky. I had topics to talk about before you people, right? Before I listened to your thesis.
yeah, no, and it was great. I remember, right, you faxing me back a few things. And I remember, so not only was it the experiment, but I thought that you had one of the first, where you modeled it, where you did the mathematical modeling of the susceptibility effect, which was, I thought that was one of the nicest papers, one of the most beautiful, not only you had the convincing results, but a convincing model. And I think Minnesota did something also important that helped establish the contrast, which they varied the echo time, something very simple. And that showed that the signal decreased with shorter echo times, proving it was a susceptibility effect. And that was also a really nice thing. And I remember, looking at your modeling paper, that was another chapter of my thesis. I thought, okay, I'll try to build on that. And I think that it was nice. Obviously, and I remember talking also, it was nice how also open and collegial everyone was, and still are. But back then, I remember seeing Tom Brady's work, and I remember calling up Robert Weisskopf, who, yeah, now he's like he dropped out of the field completely. Now he's a finance person on Wall Street or something. But he back then he was the MR physicist. He was one of the best MR physicists in the world, and developing echoplanar imaging. I remember asking him like, okay, what was the echo time? What were the parameters? and so he helped me get our first results with Eric Wong, who obviously was critical. But and yeah, I remember talking to you, getting the thesis, and then but then after that, so I think that Minnesota and MGH, I mean, it was competitive, but it was also sort of a friendly competition. I mean, it was sort of, and everyone had a sense that this was really important, overwhelmingly important.
Mm-hmm.
Did you have that sense? Like when you worked with MGH, it was exciting. Did you have a sense of how significant it was? And
Oh,
yeah,
the meaning of the result, what could be. Yes, the simple idea. I don't have any specific idea. Well, if you can look into your brain without opening your skull, and if it is saying something which is active there, that's a great thing. And
yes,
was only people knew is that for the patient when they open their skull they can measure electrode response, yes, but you
yeah,
cannot do normal people to study that
yeah,
kind of non-invasive
yes, yes,
ability of MRI. It's great. That was a major part of my. I thought this is an important part of the field of that. However, I didn't know like resting state signal you people did, or those various network formation, how they are important they are.
yes,
Actually, in the early days, I was a little bit the signal when you see in your motor or visual, they are large, so you can see very easily. But if you go into much more higher, sophisticated location, signal is not as big. So
yes,
I was worrying about if that is too small, they cannot
yeah,
do any experiment. But
yeah,
many people are very smart to to deal with those weak signal with many statistical analysis. Yes, which
yes,
I didn't expect so much. Well, yeah. But
yeah.
anyway, yeah, many people did so good, including you people. Anyway,
Well,
no, thousand of MRI oriented neurologists. They're working so hard, right? In the before
Yes.
2000 and after 2000, etc.
Oh yeah.
Enormous. Yeah.
It's yeah, now there's oh my gosh. I mean the field, I mean it's exploded and I think that it's exploded in terms of understanding the brain. It's exploded in terms of the number of people doing it. And you know, obviously people are looking at clinical, trying to understand clinical conditions. The one thing that still I think that people are concerned about, not really concerned, but I mean, I think that we're still hoping that it will become more clinically useful. And that's hard. That's a hard thing. I think it will, but I think that's where initially there was this excitement. NIH sort of has given millions and millions and millions of dollars for grant money for developing fMRI.
Yeah.
And but the hope then is that it'll become more of a daily clinical practice. Like you can put a person in the scanner and say, "Oh, you should get this treatment," or and that hasn't quite happened yet. It's not because fMRI, and that's what I tell people. It's not because fMRI is not sensitive. It's because we're understanding that people are more variable than we expected, and so it's hard. And so, what do you think of that? Do you think that fMRI?
Well, the most important point is neurovascular coupling. There is there. They really do well without failing too many cases, right? It's amazing. That's
Yes,
why that means the brain need blood so much, I guess.
yes,
One of the reasons they are responding so well, that may be the reason. But anyway,
yeah,
but those things are a very strong point. When I did early days, only thing I had in mind is 100 years old neurologist experiment, right? When
yes,
you excite something, it changes in the brain. That's the only thing we did. But anyway, that was in the later years that those neurovascular coupling molecular mechanism or whatever structural mechanism is being studied well. Yeah,
yeah.
but still there are one question. Probably you have answered to me.
Okay.
The neurovascular coupling. How many types there are? Like astrocyte to the coupling holding the capillary entrance or whatever it is. Well, what
Yes,
else? What else means neuron itself, right?
yes. There's
Neuron
neuro.
is also part of these things. And
Yes,
the way the optogenetic people did the experiment really well, only when they inhibit those astrocyte activation, they see by opening the neurons, they see the both signal.
yes,
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 that means body, body itself can certainly has a capability to open the blood vessels,
yes, yes. Which
right?
is interesting. And we're
Yeah,
still, and actually, you bring up a good point. I mean, it really catalyzed that research, like neurovascular coupling research. And I think that you know there's many wonderful papers by Atwell and you know trying to understand sort of metabolism and neurovascular coupling, and we still don't understand it. You're right, we still don't fully understand it. In fact, I think a group of at Yale, partially some of the authors were at Yale, Rothman. They have a new paper out. I think it's 2 years old now, suggesting, I think, and this is my interpretation of the paper, suggesting that increase in blood flow is not necessarily to deliver oxygen. It's more to remove waste, to remove the CO2, keep the pH constant. And so now even the purpose of the flow increase is being questioned again. So
well,
I don't know.
that's okay. If you really want to know the quantitative, what that means, what the signal change is that much of MRI signal correspond to what physical phenomena changes, something like that. That is question we can't answer. We shouldn't ask them. That's the thing.
Yes.
Even you under microscope, you see the blood vessel changes, by activation. Yes,
Yes.
and
Yes.
that is okay, but then, well, never mind.
Ah, no, 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 no. I keep on going.
Okay. Uh,
Yeah.
anyway, yeah, that's great. The reason I am saying this kind of thing is, John Hopfield, who got the Nobel Prize this year, on the physics. I know him very well because he came to the lab to get into biophysics instead of solid state physics. So when I was talking to him, I told him I like to know those activities site what they are talking each other. He said
Yeah.
he
Yeah.
sent me a picture of hundreds of birds flying in the sky, just running like this. Well,
Yes.
you can't expect you can find any of the information from each brain of the bird.
Yes. That's
But yeah,
that's a good point.
I was not asking that question, but I thought there is something important things in there. But I still have that idea to do the experiment. But unfortunately, Japanese government don't give me any money on that one.
So what experiment would you think would that you would like to try? What
I cannot tell you. I have told my friend to the experiment. He is starting to do it, so I cannot
okay.
go there.
Okay. Well, that's fair enough. That's
In a year or so, probably we can tell you.
okay. Okay. All right. Great. So trying to understand
Sorry, that's a joke.
yeah, no, no, no. That's totally understandable. But I guess the purpose is to understand how neurons are coordinated, our brain areas are coordinated, kind of like you're saying, like the starlings flying in the sky. Or okay, okay. Well, yeah. I mean, that's the thing about fMRI is that what's beautiful about this is that you can test these hypotheses, and once again, the answer might be there in the data, but you just have to know how to look and how to make the connection to a
Mhm.
deeper model. And it seems like that's what you've done. So yeah, so along those lines, along the lines of also neurovascular coupling and bold effects, there's been much other work. I mean, I know that back at the NIH, Bob Turner obviously was also doing early studies with cat brains, but also he's working with Denis Lebihan. And I remember before I started doing bold, I was trying to see something with his IVIM model, his intravascular dephasing model, basically saying that perfusion is like fast diffusion, and then but that didn't quite work. And now Lebihan is a proponent of looking at diffusion changes like cell swelling diffusion changes. And of course, there's many other groups, some from Korea recently, that have claimed more rapid neural activity that's detectable. Not sure exactly what the contrast mechanism is. What do you think of these? Have you thought about other brain activation contrasts? I'm optimistic but skeptical at the same time. I think nothing is really going to compare to the sensitivity of bold, but and bold is really useful.
You're not talking about the our experiment with activation one by two activation together or something like that nature. That's all.
Yeah, well, no, no, not that one yet, not that one yet, but more just like other contrast mechanisms than bold,
Yeah, yeah,
like direct neural current, like the fast neural currents or self swelling or things like that.
yeah.
I'm wondering if you thought about that at all.
Point of that experiment is if there is any interaction of physiology, two types of physiology getting together, then the Bohr signal can change.
Yeah. Oh, yeah. So
Yeah,
that experiment. Okay, let's talk about that. So I love that. That was my favorite, aside from your first papers. My favorite paper that you wrote was looking at the interaction of two different areas, one inhibiting the other, and using the slow bold signal, but then to see these fast interactions of inhibition. And I strongly feel that is something that is still not being explored enough.
my associates have been working for years on that one, but still, I'm asking him, "Oh, you need this point to clear. That point isn't clear yet." Well, anyway, unfortunately, that phenomena we observed is just occurs only relatively restricted condition. Some part was not excited before, then you excite it, then next one comes, the field potential goes down, that cannot excite anymore at that time. After a certain time, it recovers there. That's okay. But if you do repeat many times, they're getting into some steady state. So only that
Yes.
kind of part of the inhibition, or whatever you call it, that kind of relatively not so interesting phenomena itself, because of the once you're excited, those charges getting back to the normal condition on many neurons, it takes couple of tens of milliseconds, something like that nature. That's what we
Yes.
observed. It. But if that is important for the physiology of the system, then it's
Yes.
worth looking into it. But I don't know what
Yeah.
else. If you have some one input comes in certain area, but if another input from the side, it may interact each other. That kind of thing is there is very important, but
Yes.
it's very hard to follow yet. Yeah.
Yeah. And just to for the people listening, there's a paper that you wrote. So the title of the paper is it's in PNAS, an approach to probe some neural systems interaction by functional MRI at neural timescale down to milliseconds. Yeah, that was a PNAS paper. It was in 2000. So, and I think that, and what I liked about that, so you're right, the experiment was the main experiment was, and I think it's worth talking about a little bit more.
Okay, I'm very much interested in
Yeah,
what
it was
is
a
it?
Yeah, it was a two rats. It was a rat forepaws, and you stimulated one forepaw, stimulated the other. If they were far enough apart in time, then both bowls were the same. At right about 50 millisecond delay, one bold signal became attenuated, and then if they came closer in time, then it went back up again. So there was a tuning curve of like inhibitory interaction. And I actually think that the same thing happens with the visual cortex. Why 10 hertz is somehow the peak frequency, the peak activation, because you might have a stimulus, and then 200 milliseconds later, there's inhibitory
Yeah,
input,
yeah.
and then it reaches a steady state. And I think there's so much, all those transients we
Mm-hmm.
see in our bold responses
Mm-hmm.
is probably that.
Mm-hmm.
And so that's all this inhibitory
Mm-hmm.
activity that we haven't really understood and mapped, and we can, I think,
Mm-hmm.
with this technique. So
Yeah,
I like it. I
yeah,
think it's
yeah. If the two things getting increase the activity, not inhibiting it, that's a great discovery if anybody has seen it. Probably
yes,
physiology, there are many physiology like that one, right?
yeah,
Yeah.
yes,
If it happens around the neurons and the blood vessels, we see that both signal changes like that way, in that sense. That's very
yes.
interesting. Yeah.
Yeah, I do think that that's a wide open
Yeah.
sort of area that's
That's
worth
right.
probing.
Yeah.
I totally agree with you. I love that paper, and I still bring that up to people in my lab, and it's that we should think of other types of experiments to explore that
Mhm.
effect.
Mhm.
But I guess what I was also asking about is, so aside from that, what do you think the future of fMRI is? Do you think it will be clinically useful? Do you think it will be just a tool that we can actually use in complementary with EEG and
Yeah. Yeah.
other techniques? Yeah.
Yeah. I have been asking people question in a way why fMRI is not strong enough to be clinically useful. Why is it? Probably you have an answer to it.
Well, I actually think it's because that the brain, that people's brains are variable enough it's not like it's like measuring blood pressure. Like, blood pressure, it's not, if you have a blood pressure that's high, you know that you have a problem. But with the brain, it seems that if you have, let's say, schizophrenia or something, there might be different manifestations of this, and there's enough variability that we haven't figured out how to put one person in the scanner and say, oh, this is what we need to do, because I think that we don't have we don't understand the disorder well enough to do that.
Yeah.
So, and I think that, so when we look at the activity, we see all this stuff. Some is similar, some is different. It's even different among people who have different types of schizophrenia, or
Yeah.
even the similar types, and that might look different. And
That's
so
right.
I think it's because people are different, and we're still getting, and a big trend in fMRI now is to sort of understand those individual differences. And I
That,
think
that,
that will allow
that, is
clinical.
it. That was the point I was trying to make to you now. The reason why it is not so reliable is because single person's fMRI data, analyze it many ways, is not reliable to the clinicians. Same experiment, they did the same person, it looks different. It looks different. But that is, if you make your data, individual single person's data is completely reliable. There may be different at this time and the other time. You maybe find the differences in many ways.
Yes,
Only point is, so far, probably you people have done on a very good point on that kind of problem to make fMRI data more reliable. Since I cannot do any more experiment, so I will sit down on the screen doing those MATLAB work to make it possible or not. So far, I am not successful yet. I'm
uh,
still working on it.
yeah. No, I think we need to understand. So it actually it's really an important point that, as you say, it's not reliable, but it's not because fMRI signal is not reliable. It's because brain activation is not reliable.
I see, I see. Okay, that's another question. That's
Yeah,
another question. Yeah,
yeah. And I think so. It's not a criticism of fMRI. It's sort of a statement of, people are more different than they vary more than we think
yeah,
they
yeah,
do.
yeah, yeah. Yeah, but once you get your data reliable, then you can ask that question. Before
Yes,
that, you cannot ask that question either. The one
yes,
side to the other correlated. That's not reliable, the correlation anything. Okay.
exactly.
Yeah.
Yeah, I completely agree with you. And that's a very good insight, and I think that more people should understand that too. That this is what we're struggling with. This is and fMRI can be used clinically, and I think, it's pre-surgical mapping or things like that, but it hasn't quite caught on. And I think once it does catch on, then all the big vendors will start making better fMRI platforms
Uh,
for doing it. And that will help that will be a catalyst for more change. But yeah, so over the years when you've been doing these experiments, so how did this, I mean, obviously this discovery of Bold changed your career. I mean, you know, it's funny, when I was getting my PhD, I just thought, oh, good, great, I have a PhD project, but it obviously my whole career has been committed to this as well. But has there been anything that has surprised you most, or has there been any discovery with bold contrast or other sort of result that sort of was surprising or disappointing, or is there anything that stands out?
well, unfortunately, my brain is so poor those days. That's why I don't read too much. All other papers in the world, right? I don't know anything of those kind of things recently. That's why
Well,
I'm
that's true.
concentrating myself on that particular question. That's the only thing I can do.
Yeah, no, it's hard even for people who read a lot. I mean, you can't keep up with it. I can't. I like to try to keep up, but it's like it's a lot of work, a lot of papers. It's funny because and it may be related to the clinical applications because obviously when people like Ravi and myself get together at meetings, we talk about things and obviously it comes up, when we mention your name, we always say, oh, well, and I don't want to push this too far, but we always say, oh, well, Seiji, he he deserves the Nobel Prize. You do. And and I think that I don't know, and we were always mystified, like why this is a major discovery. And I think maybe because it hasn't quite caught on clinically enough. I don't know. I don't, it's just, but you don't have to say anything to that. I mean, obviously, this is what we say about you. But,
Uh,
um,
my friend, John Hopfield, who got Nobel Prize on his neural network analysis, right? Up to
okay.
up to last year, probably he never expected he got the Nobel Prize. The reason why he was got the Nobel Prize is because many of those neural analysis or AI treatment of many things
Yes.
often based upon his neural network analysis type. So he did a very important contribution. Yes,
Yeah. Yeah.
unfortunately, fMRI doesn't go that much high level. Yeah,
Right. Right. Not
yeah.
yet. Not yet. That's we're all trying. We're all trying. And I think that it will. It's just that it's interesting. I mean, fMRI has gone in stages where after your discovery, then there was, resting state fMRI, which was a big discovery. And then then it went into things like big data, like large data sets, people analyzing massive amounts of data for, trying to get biomarkers. And that's sort of, I feel like people now have taken a step back and said, well, we have to rethink about it. I think right now it's in a new stage where we're trying to understand individual variability. And I think that has the most potential for having more impact, sort of understanding how people are different and similar in many different ways, which will have more clinical impact. So hopefully the impact will be high enough and the powers that be. But then again, it's not something that we think about it. You probably don't think about it, but um, so what are you doing these days? As far as you've retired, so you worked at Bell Labs for 33 years. Then you had a number of you had the Ogawa laboratories that you worked at. Then now you're retired. So how do you spend your time now?
How I spend the time sitting in front of the
What's that?
screen. When I make a single matter of problem, a project, or whatever function, I spend so much time because I always make mistouch on the typing, and also wrong things to do in the programming. So it takes so much time. But once you can see something, that's very exciting. So one day
Yes.
I got a certain result. It was so exciting. Now we should do this. So I told my friend, why don't you try this one?
So
And
what are you working on? You're programming up stimuli or things like that, or what are you doing? Uh,
no, the analysis of the fMRI data,
okay.
bring it to them. So
Yes.
he said, okay, but before, see, few days later, I look into my result because I wanted to make sure before this getting public, I should be sure this is right or not. I did a phenomenal, fatal mistake in my program, so that I have to call them up immediately.
Yeah.
Anyway, so that I'm spending time just sitting on the with my PC, looking under the big screen.
So are you spending time? Are you
Not,
processing data?
not as sophisticated MATLAB approach, but very old-fashioned slow approach. I'm spending time like that way.
Well,
Yeah.
sometimes those are the best approaches like that, though. I mean,
Hmm.
I still think sometimes that's the best. Uh, Sometimes those approaches produce results
That's right.
when, you know,
Yeah, more or less like that way. Your Python approach, Python way of doing things, I haven't run that way, so I'll try to run sooner or later. Looks much a better way of doing it, I guess. Okay.
well, that's great. So you're learning Python. You know Python. So you're programming in Python
No, no, not
analysis.
yet, not yet, not yet.
Okay.
My associate may have it, but I don't have it.
Okay.
I will try getting into it.
That's amazing.
Even
That's
though
amazing.
I am a little bit too old to start as something new, but
I have to remind the audience that you're 90. You just had your 90th birthday, which is
yeah.
you're amazing. You look amazing for. I mean, you look no more than 75. You're taking good care of yourself. That's yeah.
So physically, I am a very good condition, so that I can keep doing this for some years to come, I hope.
I I I hope so too. I hope so too. I think I was talking to Ravi Menon, and he mentioned that he talks to you. He mentioned that you play tennis like 3 times a week as well. You play tennis.
Yeah, yeah, that's right.
Okay,
That's
okay.
a, that is a in my schedule. It is a must
Okay.
day. You cannot commit to that way. Although
That's
I'm not so good tennis player, but some of the young friend, young means 70s or sometimes beyond the 70s. I'm just competing with those guys, doing so
that's
far okay.
that's great. That's great. So I was still wondering. So okay, so now you're working on just it's interesting that you mentioned that you're working on sort of trying to find ways of just processing the data that are unique that pull things out. Also remember Jim Hyde as he was approaching, he still came into the lab like once a week after he retired, and he would do the same thing. He did
Did
the
I?
same thing.
Just
He
a little bit sound. It's not. It's very hard to catch your voice.
Oh, okay. I'll try to speak up.
Yeah,
Maybe it's my microphone.
yeah.
Um,
Make it a little slower, then I can catch.
okay.
Okay. Okay.
So I remember Jim Hyde also uh, you know, towards after he retired, he was also doing like
Uh-huh.
working on ways of processing data and,
Yeah.
just little thoughts, like a thought, oh, let's try this or let's try this. And that's interesting.
Yeah.
So let me ask you just a few more questions. Um, and sort of we kind of went over this during when we were talking, but uh, so I'm wondering. So when you discovered uh, Bold, and what do you think was the most important thing leading up to that?
The important thing is what you should know what is important. You can't
Okay.
spend unimportant trivial things, even though you are funding required to do it. But anyway, if you have some project, you have to find what is important for that. Or even you choose a project, you have to know what's important to choose which project to work on. What is the importance in there? That kind of question that will
Yes.
give you what to do often.
Yeah.
Once you know what to do, what is important, then you should know how to do it. What is to be done? What kind of approaches or attitude will be the very important thing to do. That was I
Okay.
learned from Howard McConnell.
Okay. So how did you know? So then the question is, I'm sure people listening, how did you know that doing this was important?
That is, very soon I switched from spectroscopy to imaging. So I
Yes.
wanted to know, imaging is so powerful things. I knew I was impressed with those images, but my point was what those image signal itself to make the image. What is important? What is going on behind the signal? I like to know what the signal means in some sense. T1,
Yes.
T2, yes, it is, but something more.
Yes.
The T1 probably very hard to change it. T2 is easy to change.
Yeah.
Even real T2, it's easy to change. It depends upon how the water is bound, something like that nature. But
Yes.
anyway, I wanted to know what things behind them. That was therefore I went into very high resolution images. Then
Okay.
I encountered some of which I didn't know. There is a present.
Yeah. And so so then the question would be, uh, so then everyone today, like there's so many researchers working on fMRI, and everyone is, they're trying to find an important, what's most important to
Mhm.
work with fMRI, and maybe it's something else. Maybe it's some aspect of fMRI. Maybe it's hard to know ahead of time what will be important. I guess that takes a certain amount of intuition and experience and
Yes. Well, to find what is important is your people's job, not my job anymore. I'm a little too old to make new things.
but yeah, no, I agree. I agree, and it's hard because it says you knew what was important and also what seemed like it was achievable. Like you felt, okay, there's a mystery here, but I think it's it's understandable if you do if you with your approach. And so that was, yeah, it's a hard, it's, I mean, obviously if it were easy, many more people would be doing it, but
Yeah.
um so, and I know I maybe asked you this, in a different way, but do you have any, like, what do any memories pop out? Do you have any memories from when you developed fMRI stand out as the most, having the most fun or the most interesting or the most exciting?
That's right.
Uh,
Well, to be able to measure brain activity by NMR, very exciting thing to do. And the prospect for the future, you may not be able to imagine at the start how far it can go. But
yes,
anyway, it has some potential there. That's what a very exciting field to come up. That's right. Yeah,
yes,
maybe but many of those initially exciting and promising new phenomena finding, but doesn't last too long. Many things doesn't last too long. It is very interesting, very important, but doesn't last too long.
that's
In that
true.
sense, fMRI is amazing. It
Yes,
lasts so long. So
yes.
many people working so hard, still finding meaningful things, I guess. Yeah. I understand your contribution to that direction is very good, very important you have done. Yes.
Well, we do what we can, and I think I think the reason why fMRI lasts so long is because, right from the start it just worked. I mean, it's really robust, and there's still, it's such a strong signal that there's so much information still in it that I think that we're still trying to figure out how to pull out,
Mhm.
and that's, I think, what keeps it going.
Yeah.
And also, of course, what was really useful with what I like to mention is that, when you discovered Bold and people discovered fMRI, there were already scanners all over the world ready to go, ready to do it. And it just, they just jumped on the clinical scanners, and that helped a lot get it going, and it's keeping it going.
Yeah.
It's sort of, yeah, it's relying on clinical MRI being successful. But
You have many way of doing brain science, I guess, and they are very much sophisticated, and maybe measurement itself much more specific than the board, but
yeah,
still okay. But merit of the board is very simple to measure. That means you can put the brain in many various conditions.
yes,
Unless if the brain doesn't move too much, it's a great way of doing things. That's what
yeah,
brain works way. The way brain works. That's why one of the advantage of or benefit be able to do in non-invasive such a device and way of doing this.
yes, yeah.
So
I, that's
therefore, still there is a room to grow. That's what
yes.
I think.
I completely agree. I completely agree. And
Yeah.
um, but okay, so I guess oh, let me just ask you this. So you spent after Bell Labs, you spent a lot of time in Japan. I mean, obviously your home country, obviously. How is what is research? How is research different in Japan than it is in the United States? Is it like, especially for fMRI, for instance? I know of many really outstanding groups like in Osaka and Tokyo. There's Riken. There's several other groups. Is how does it compare? Like the environment for doing research?
Well, Fukunaga is in charge of those things. You know Fukunaga, right? He's in charge of the field of functional MRI, whatever it is, and also very high field things. They have gotten new 17 again, and
Yeah,
also they're getting a very high field animal machine, I guess, something like that. And
yeah.
his institute is open for everybody to come and work with them. That
Ah,
that's great. Yes, my associate goes there too, to
oh,
working
really?
with Fukunaga. Yeah, and
Okay.
well, there are other group who are more centered on the human activity in the brain. That
Yes.
I haven't gone too far to see how they are doing nowadays. And
Okay. Okay. It
see,
seems like it's growing.
I don't
Yeah.
know. I don't know how far different from in the world, like your place, and also in the many sophisticated research area in States and also in Europe. Yeah, so
Yep.
many people, many good people working, I guess. And
Yes. It is amazing, and it's not. It's we're lucky that our governments still support it, and they support and they want to foster. They see the potential of
mm,
this, which
mm,
is
yeah,
large.
yeah.
Um,
I unfortunately I am a little bit too old to. Recently, I haven't read the journal at all. I don't know what's going on there. I always ask my collaborator there, what's going on these topics?
I think it's become so big that nobody knows exactly what's going on. I think even if you read 10 papers a day, it's hard to know what's going on exactly. um, yeah, and I think so. Right, that's a struggle we all have, and but I think actually, what's I think a lot of people who do some of the best work, I think they sometimes you can get too aware of what's going on and sort of and not really. And I think sometimes there's something to say for knowing the field, but also just sort of focusing on your work and doing your thing as best you can. I think that's yeah. It's hard to.
Yeah, I wish I could have done a little bit more advanced topics rather than the initial stage of function MRI.
Well, like we said before, I think that you set the groundwork, but also I think that your idea with looking at the interaction, the cross interaction, and based on the timing, and maybe based on suppression or enhancement,
Mhm.
I still think that idea that you
Uh-huh.
had
Yeah,
is
yeah,
good.
that's right. That's right. It is a little tough experiment. You have to choose a timing, and also signal may not be as strong as you expect. If you go to a higher region of function, it's getting weaker in that sense. But
Yes.
even so, very measurable. And
Yeah.
also, our associate published a paper with my name on that too, on the timing cross-membrane, how fast cross the left to right hand side the brain, timing-wise. 15 milliseconds, what kind of timing we thought we are measuring it, but yeah, still lots to do. It's not
Yes.
necessarily clear-cut results. But, mm.
Yeah.
Yes, if you're lucky, you can find somewhere very important information getting it together at one site and how they are interacting each other. That kind of thing is if you can find it. It may not be so easy, but something there is a possibility there. Yeah,
Yes. Yeah, and I think it might open up other things like,
yeah,
understanding like inhibition or
yeah,
other things in the brain. So, that's still we're working on that. So, at
yeah.
least my group, I'm thinking about that. So,
If you
um,
can find some such a phenomenon, not on the inhibition side, but more the excitation side, then maybe it's much more interesting too. There must be
yeah,
there, right?
that's
Exciting instead of inhibiting, it should be improving the changes, signal changing, increase or something. More
yes.
signal coming into because of the two inputs colliding each other. It's maybe there must be there too.
Right. I think that's doable. I really do. And
Yeah,
maybe with layer fMRI, there might be some hope in looking at cortical layers and,
that's right.
output versus input and combining that with the
Yeah.
modulatory effects. I think, yeah, there's work to be done. Yeah.
If
Yeah.
you rely on the neurovascular coupling, both signal may be the thing to use for exploring those kind of new approach. Other technique
Yes.
is so sophisticated, it's hard to follow new.
Right. I think that the fact that bold sort of integrates over time, it allows us time to look at integrated effects. I think that's a good. Sometimes it's a good thing as opposed to just because like you said, the neural interactions are so rapid. Um, yeah. Sometimes, I mean, that information is useful, but I think with bold contrast, I think the integration over time of the effect is
Mhm.
also can be
Mhm.
more useful. People always look at it as a detriment, but it's also sort of useful sometimes. All right. Well, I think then we've covered a lot of ground. Between 2 days ago and today, we covered your history, main findings. I'm trying to understand. Like what came out, what how you when you
Mm.
discovered gold and what you're doing today. Well, thank you very much, and
Yes,
thank you.
that's nice talking to you. It's very
Yeah,
lovely.
thank you. Thank you. All right.